everyone. I'm Vincent Racaniello. Welcome to Session 3 of Week 8 in Virology Part 1. And in this session, we're going to be talking about how viral transcription may be regulated. There are, of course, specific proteins that regulate transcription. We've talked about the regulatory sequences that surround the promoter. Viruses in general require these host proteins or virus-specific proteins to regulate specific promoters. In addition, viruses can either encode or bring in with the virion what are called co-activating molecules. These are proteins that do not bind DNA directly, but they can modulate transcription. And many of these, but not all of these co-activators, modulate the structure of nucleosomal DNA templates. Now, you should probably know that in eukaryotic cells, DNA is present in a very specific structure called chromatin, in which the DNA is wrapped around octamers, that is 8 copies of different histone proteins, in this fashion shown in this diagram. So each of these discs, if you will, consists of 8 histone proteins. The DNA is wrapped around them, and these are very near to one another. So the DNA is present in this very ordered and regulated structure. The chromatin, or the histones, can occur with the DNA in a closed or an open structure. The structure shown here is closed. It's very difficult to transcribe this template when it's closed. We now know that activation of transcription of specific genes is accompanied by the opening of the chromatin, and this is very specifically done by modifying the histone proteins by adding methyl or acetyl groups to them. So the co-activators participate in transcriptional regulation by modulating the structure of the nucleosome template, making sure that it's either closed or open as needed. Now, not all viral genomes are present in these nucleosome-like structures, but a number of them are. It's quite evident, for example, that histones are involved in the structuring of certain viral DNAs, and therefore histone modifications can affect transcriptional activity. Sequence-specific transcriptional activators are regulatory molecules that are composed of multiple domains that contribute to viral gene regulation. And if you recall, in the previous session, we showed the promoter regions with binding site for specific proteins, and these proteins are the sequence-specific transcriptional activators. They bind that specific sequence to help stimulate transcription. These are typically modular proteins, as shown at the bottom here. They have DNA binding domains. They have a domain for formation of dimers. These are usually active as dimers. And then they have an activation domain that is a part of the protein that is involved in stimulating transcription. They can also have other domains which aren't shown on this diagram, such as for interacting with other proteins. And on this diagram at the bottom here, underneath each type of domain, so there are 3 types of domains in this proteins, are the different kinds of proteins within that domain. So, for example, there are 3 different kinds of DNA binding protein domains listed here: zinc finger domains, helix turn helix domains, and basic domains. For dimer formation, there's a leucine zipper. And the activation domains can be acidic, glutamine-rich, proline-rich, or isoleucine-rich. So this is the way many sequence-specific transcriptional activators are built. Here's an example of one of these transcriptional activators binding to DNA. This happens to be a transcription protein called AP1. It is, in fact, a heterodimer. It consists of 2 polypeptides, 2 different polypeptides. One of them is called Jun, and the other is called Phos. And these Jun and Fos proteins have a leucine zipper domain, and they have a basic region which binds DNA. So what we're showing here is just the structure of the leucine zipper domain, which is the domain that allows for interaction of the two proteins, and the basic region of the two proteins, which allows binding to DNA. So the DNA that the protein is binding to is shown here, and you can see each of the monomers of the AP1 heterodimer is binding in a major groove of the DNA. These are sort of thought as forceps. This protein forms, if you think about it, a forceps-like structure that helps the protein bind snugly into the DNA. There are a variety of general sorts of strategies that viruses do when they infect cells, and they're listed on this table. Some viruses use entirely host proteins for the transcription of their mRNAs. And an example of that are the simple retroviruses, or we should say the retroviruses with simple genomes. These are viruses that produce a DNA copy of their RNA genome. The DNA then integrates into the cellular DNA, and it's transcribed by cellular RNA Pol2 using all the cellular proteins needed for that. So they only use host transcriptional components. There are some examples where the virus makes one protein and the rest of the transcriptional machinery comes from the host. And bacteriophages T3 and T7 do that. And in this case, the viral protein that is made is responsible for transcribing late viral genes, that is, genes expressed late in infection. And then the complex retroviruses, parvoviruses, papillomaviruses, and polyomaviruses make a single protein involved in transcription that is a regulatory protein. We will talk about the polyomavirus regulatory protein a bit later. And then the adenoviruses, phage T4, and herpes viruses use the host transcriptional machinery plus several viral proteins, more than one, several viral proteins that act sequentially to stimulate transcription. And we'll talk about those in the context of adenoviruses and herpes viruses. Now, there is one exception to all of these other viruses, and that is the pox viruses, which encode the entire transcriptional machinery on their own. They don't use host machinery at all, and they do all the transcription of their genes in the cytoplasm. There are two general patterns of regulation that occur during transcription in virus-infected cells, and these are called regulatory circuits. And they are established by proteins, these regulatory proteins that interact with the polymerase to establish these circuits. And we have what we call positive or negative autoregulatory loops. These are two variations on a similar theme. The positive loops, of course, alter the rate of initiation. They increase it, and these are modulated by proteins that stimulate transcription. The negative autoregulatory loops involve the production of proteins that repress gene expression. And then we have the second form, or the second pattern of regulation, which is a transcriptional cascade. And here, transcriptional units are activated in a fixed sequence. So let's take a look at an example of both of these patterns now. So on the top is an example of either a positive or a negative autoregulatory loop. This one happens to be a positive, but negative would essentially be a slight variation on this theme. So here we have a viral DNA which has entered the cell. It is transcribed at a specific promoter to produce protein X from gene X, and of course the entire transcriptional machinery is used to produce this mRNA. The viral mRNA is translated to a protein, and this protein X happens to be a transcriptional activator. It stimulates the promoter, so it goes to the promoter and becomes part of the promoter sequence along with Pol2 and all those other cellular proteins. So this is what we mean by cellular components here. We're talking about the cellular transcriptional regulatory proteins. Protein X becomes part of the promoter initiation complex and stimulates transcription. So now we get lots of gene X mRNA made. And again, that is made mainly by cellular components with viral protein X. So the idea here is that protein X stimulates transcription beyond what the cell would do on its own. And if this were a negative cascade, you would imagine that the protein, instead of stimulating transcription, is repressing it. Maybe it comes into the cell, the viral genome comes into the cell, and this promoter works at a high rate. And then, for whatever reason, it needs to be ramped down, and it's done by producing a negative regulator. So they're autoregulatory because the protein produced by this transcriptional unit represses its own mRNA. The second part on this slide illustrates cascade regulation. Now here we have again a viral DNA coming into a cell. Cellular components lead to the production of a viral mRNA transcript, which in turn is translated into protein X. Protein X now goes and activates the promoter for a different gene. Not the same one as in autoregulatory loops, but a different promoter. And that then results in the synthesis of gene Y. So gene Y mRNA synthesis is dependent on protein X. It cannot occur without protein X. So this is cascade regulation. So again, gene X can be transcribed only by cellular components, but gene Y cannot. Gene Y requires protein X, so that's where the cascade comes in. You have to make protein X first before gene Y can be transcribed. So it's very different from the auto or regulatory loop where gene X can be transcribed just by what is present in the cell. Viral protein X, in the case of an autoregulatory loop, simply enhances or represses transcription. So let's just list a few of the viral transcriptional activators that we know about, just so that you know the players when we mention them. We're going to mention some of them in this week and also later on in this course. So there are some autoregulatory molecules, like the T antigen of SV40. We talked a lot about that during our week of DNA replication, and now you see that it can also regulate transcription. The protein of herpes simplex virus called ICP4 is another autoregulatory molecules. Some of these transcriptional activators bind DNA. The T antigen does. An EBNA protein of Epstein-Barr virus, the herpes virus ICP4, and the adenovirus E2 protein. All these are DNA binding viral transcriptional activators. Some of them also bind host proteins, the herpes simplex virus VP16, and others liberate host transcriptional activators like T antigen of SV40, the adenovirus E1A protein, and the E7 protein of papillomaviruses. So different ways you can be a viral transcriptional activator. In general, there are three kinds of viral transcriptional programs, as illustrated here. And we're using the genomes of three viruses to illustrate them: SV40, adenovirus type 2, and herpes simplex virus type 1. These genomes are not drawn to scale because, as you know, this SV40 genome is much smaller than that of AD, which is in turn smaller than the herpes virus genome. The simplest transcriptional program is illustrated by SV40, which has a small genome, has only an early and a late phase of mRNA synthesis and replication. As the genome comes into the cell, the first protein that is made is the large T antigen. This is made initially by transcription using cellular components. The large T, as you know, binds to the origin and stimulates viral DNA synthesis. And it also stimulates late promoter activity. So the viral transcription program is run from early and late promoters. The early promoter works without large T, and in fact, eventually the early promoter is repressed by large T, favoring synthesis of late mRNAs, and those encode the structural proteins. So it makes a lot of sense to regulate in this fashion. You don't need the viral structural proteins early on. You need to make proteins that will replicate the DNA, and only later do you need to make capsid proteins. Another type of transcriptional program shown here by adenovirus, slightly more complex. We have 3 phases of transcription. We have immediate early, we have early, and we have late transcription. So when the double-stranded genome comes in the cell, some immediate early messages are transcribed, and one of the products of those mRNAs is the E1A protein. And the E1A protein is a transcriptional activator that is absolutely required for the synthesis of early mRNAs. So this binds at the promoter and is needed for their transcription from that promoter. One of the products of the early mRNAs is the E2 the E2 protein, which is required for the late phase of transcription. It actually doesn't activate a promoter, but rather allows the viral genome to enter into the late phase of DNA replication, where you get lots of genomes and amplification of late transcripts. And among these are transcripts encoding structural proteins. The herpes simplex virus transcriptional program also has 3 phases: immediate, early, early, and late. But this is a little more complicated because the virus particle itself brings in a transcriptional activator called VP16. And VP16 is needed for the transcription of immediate early mRNAs. And those give rise to the synthesis of 2 proteins, ICP4 and ICP0, which are transcriptional regulatory proteins. ICP4 in particular represses immediate early transcription, but both 4 and 0 activate early and late mRNA synthesis. And the transition to late mRNA synthesis, in addition to 4 and 0, also requires viral replication programs. So 3 different transcriptional programs orchestrated by transcriptional activating proteins, and again illustrating that the way that you regulate the kinds of viral proteins you need, depending on what part of the replication cycle you're in, is to regulate mRNA synthesis. Let's take a look at VP16, this very interesting herpes simplex virus protein, which is actually brought in the virion. So this is required to make the viral immediate early proteins. The genome cannot be transcribed by simply utilizing the cellular transcriptional apparatus. It also requires this protein VP16. So the solution is to bring it in with the virion. So VP16 gets into the cell. It gets to the nucleus, and there it acts in a couple of ways. First, shown at the top here, we're looking at the formation of the closed bent initiation complex. Here we have the transcription proteins binding to DNA, and then the RNA polymerase II. VP16 stimulates the transcription at viral promoters, and one way that it's thought to do so is by binding to one of these transcription factors, TF2B. And it alters the conformation of the transcription factor. You can see it goes from a closed conformation to an open one. And in the initiation complex, it's thought that in the open conformation, TF2B allows the transcription of viral immediate early promoters. The second way that VP16 is believed to operate is by unwinding the nucleosome and making the open histone conformation. So normally, as we mentioned before, DNA is wrapped around nucleosomes like this, wrapped around histones, forming a nucleosome structure. It's very difficult to get to the promoter sequences when the DNA is closed like this. So what VP16 is thought to do is to open up the nucleosome by modifying histones or by participating in the modification of histones. And now we have VP16 in here. The histones are opened up, the chromatin's in an open configuration, which allows for transcription. So two ways that we think that VP16 is allowing the immediate early promoters of herpes simplex virus to function. Here's another example of how a viral transcriptional activator stimulates viral transcription. This is the adenovirus E1A protein, which again you remember is needed for the production of early viral mRNAs. It is a zinc finger protein, that is, the protein folds in such a way that it coordinates a molecule of zinc in it. And this zinc finger portion of the protein binds cellular proteins that are participants in the formation of the initiation complex. And it's thought that E1A brings these proteins into the complex, which are needed for the production of adenovirus early mRNAs. Another way that it works, that E1A works in adenovirus infected cells, is that it interacts with a protein we've encountered before, the RB, or the retinoblastoma protein. The adenovirus promoters, shown here diagrammatically, require for their activity the E2F in transcription factor, which is often complexed with other cell proteins, one of which is called DP1 here. So this E2F DP1 heterodimer is needed for the transcription of early adenovirus genes. The problem is that the RB protein is normally bound to this E2F complex, rendering transcription inactive. So the virus gets around that by making E1A, which binds RB, takes it off of the E2F complex, and now we have adenovirus mRNAs which can be produced at these promoters.